Welcome to this video lesson, Neurotransmitters Contain Chemicals. Today, we'll be going over the steps required for signals to be transmitted between neurons and discuss some examples of neurotransmitters. Neurons communicate with each other and with other cells via special junctions known as synapses. Some neurons connect only with a few other cells. Some share synapses with thousands of other cells.
Now, you might be wondering, how can signals be transmitted between neurons? You must first understand that signals typically only travel in one direction along a neuron. Keeping that in mind, think about a row of dominoes falling over one by one. The first domino in the signaling cascade is the signal that is received by a neuron's axon or cell body. This signal then triggers an electrical impulse, also known as an action potential, that moves along the axon. Once the signal reaches the end of the axon, also known as the axon terminal, it must move through the synapse to the next neuron.
A chemical synapse is the most common type of synapse. Here, an impulse triggers the release of chemical messengers called neurotransmitters from pockets known as synaptic vesicles. Neurotransmitters leave the cell and physically travel through the narrow, watery space known as the synaptic cleft between neurons. The space between neurons is about 20 nanometers. That's about 1/500th the width of a single human hair. There is more than one type of synaptic cleft. Most often, the axon terminal of one neuron is near the dendrite or cell body of another neuron or muscle cell. Less often, the axon terminal can end near a small blood vessel and secrete neurotransmitters directly into the bloodstream, or, in the absence of a specific target, secrete neurotransmitters into the surrounding area. These are just a few examples, but even more synaptic variations exist.
Have you heard of the phrase neurons fire? If you have and are wondering what that is about, we will explain it shortly. After traveling through the synapse, the neurotransmitter finally reaches the other side of the gap. Here, it most often attaches to a special receptor molecules on the dendrite or on the body cell of the receiving neuron. Once the neurotransmitter binds to these receptor molecules, it can promote a generation of a new electrical impulse, also known as an action potential. Though the release of a neurotransmitter can make it easy for a neuron to fire, it can alternatively have the opposite effect. In some situations, neurotransmitters can be inhibitory and actually make it harder for the neuron to fire an action potential and prevent the message from being carried on.
More than 100 different neurotransmitters have been identified. Each has a certain three-dimensional shape which only fits a certain kind of receptor site, similar to a key and lock. Neurotransmitters can be classified based on their structure or based on their function. In terms of function, there are three main categories. Excitatory neurotransmitters promote neuron firing. One example is glutamate, a small chemical compound that is the most abundant neurotransmitter in the brain. Inhibitory neurotransmitters, on the other hand, prevent neuron firing. One example is glycine, which acts in the spinal cord and brainstem and is involved in processing motor and sensory information. Neuromodulators are neurotransmitters that alter the strength of signal transmission between neurons. An example is dopamine, which is produced in the hypothalamus and can serve either an excitatory or inhibitory role, depending on where in the brain it is functioning. Although all three of these examples are small molecules, scientists have also discovered that certain gases, such as nitric oxide, can also act as neurotransmitters. Unlike other neurotransmitters, the properties of nitric oxide prevent it from being stored and released from synaptic vesicles, and it is instead made on demand. Additionally, instead of binding to surface receptors on target cells, the lock that most neurotransmitters serve as a key for, nitric oxide is small enough to directly enter and affect target cells.
Once the neurotransmitter or messenger has served its purpose, it must be removed in order to prevent repeated firing and allow for new signals to be received. The mechanisms that we are about to discuss remove the messenger from the synapse. The removal of the messenger ends the signal. The first mechanism we'll be talking about is diffusion. Some neurotransmitters can float or diffuse away. When the neurotransmitter diffuses away, it can no longer act on a cell receptor. Another mechanism by which signaling is terminated is known as enzymatic degradation or deactivation. In this mechanism, a specific enzyme changes the structure of the neurotransmitter. By changing the structure of the neurotransmitter, it can no longer bind to the receptor. A third mechanism is known as reuptake. In this case, the whole neurotransmitter molecule is taken back into the neuron that released it. A fourth and the last way through which neurotransmitters are removed is through glial cells. Glial cells are the most abundant cell types in the central nervous system. They support and nourish neurons. There are several kinds of glial cells, but the kind that add to work here are known as astrocytes. These star-shaped cells can remove neurotransmitters from the synaptic cleft. Now that we've learned about how neurons fire, let's quickly test our knowledge of the process.
Take a look at these steps. What is the correct order that needs to occur in order for a neuron to successfully fire using a chemical synapse? Feel free to pause the video if you need more time to think. The correct order is C. An electrical impulse travels to the end of the axon. Then D. Neurotransmitters are released from the axon. Then A. Neurotransmitters move across the synaptic cleft. E. Neurotransmitters attach to special receptors on the receiving neuron. And finally, B. Neurotransmitters are cleared from the receptor sites and the synapse. To help us distinguish neurotransmitters from other types of chemicals, scientists have created rules to help us determine which substances qualify as neurotransmitters and which don't. The criteria are as follows. 1. The substance must be synthesized in a neuron. This is one criteria that distinguishes neurotransmitters from hormones, which can similarly act on the brain but can be synthesized elsewhere in the body. 2. The substance must be found in the axon terminal or terminals of the neuron. 3. The substance must be released in response to an electrical signal in the cell membrane. 4. The substance must cause an effect when released from the neuron or when applied externally as a drug. And 5. There must be a mechanism to inactivate the substance or remove it from the synaptic cleft.
Though we have been talking a lot about neurotransmitters and how they help neurons communicate, it's important to note that sometimes neurons do not communicate using neurotransmitters. Another way in which neurons can communicate is by having an electrical charge pass directly from one neuron to another. This is known as an electrical synapse. In an electrical synapse, the neurons that are communicated are very close together. Through this type of signaling, neurons are able to communicate very fast. In fact, they communicate faster than those communicating across chemical synapses. This is because receptor binding is not required. Instead, signals are rapidly transmitted across electrical synapses. Unlike what we have discussed before, this type of communication allows many interconnected neurons to fire simultaneously. Really cool, right? These electrical synapses are really important, even though they are less common than chemical synapses. Anyways, thanks for watching. We hope you learned a lot from this video.